Have you ever really thought about how something as complex as an eye actually gets built? We're not talking about evolution over millions of years, but how it happens inside an embryo, starting from scratch. Today, we're diving into one of biology's most incredible construction projects, the development of the human eye.
So that's the big question, right? How does a simple sheet of cells know how to fold and communicate and transform itself into this intricate light-sensing machine? Well, let's trace that journey right from the beginning and watch how biology pulls off this amazing feat step by step.
All right, here's our roadmap for this explainer. We're going to look at the blueprint, see the first signal, watch the cup and lens form, then build the inner eye, and finally add the finishing touches. Let's get started.
Okay, so first things first. Before you can build anything, you need the right materials. For the eye, it all kicks off with one very specific layer in the super early embryo.
So, in a tiny embryo, you've got three main layers of cells. But for our story today, we can pretty much ignore two of them. We're zeroing in on just one, the ectoderm. This is the powerhouse layer that gives rise to our skin, our entire nervous system, and, you guessed it, our eyes.
Now, let's get to the action. We're at the point where we see the very first visible sign that an eye is starting to form. And what's wild is just how early this all happens.
And check out this schedule. This is not a slow, casual process. It follows a super precise timeline, kicking off at just day 22. That's only 3 weeks into development.
So you can almost picture it, right? The developing brain starts to send out these two little outgrowths, like it's reaching out towards the surface of the embryo. This is it, the very first step in building a bridge between our internal world and the outside world of light.
Okay, this next part is maybe the most exciting and dynamic phase of the whole process. It's all about a moment of physical contact, a sort of crucial handshake that sets off a chain reaction you just can't stop.
And what's so cool here is how direct it is. The cause and effect is crystal clear. That little bump, that physical touch, isn't just random. It's a signal. It's one group of cells telling another group, okay, your turn. You are now officially destined to become a lens. It's a classic example of what's called inductive signaling, and it's fundamental to how we're built. Now, in response to that signal, the vesicle starts to fold inward on itself. This folding process is called invagination, and it's a key mechanical trick that nature uses all the time. Here, it turns a simple ball of cells into the fundamental cup shape that will become the back of the eye.
And look at this beautiful coordination. While the optic vesicle is folding inward, that new lens placode does the opposite. It dives in from the surface to meet it, eventually pinching off completely and becoming its own little sphere, perfectly nestled inside the cup it just helped create.
So, let's take a breath. We now have our two core components in place, the optic cup and the lens vesicle. From this point on, each one starts its own specialized construction project to build out the eye's internal machinery.
Now, those two walls of the optic cup, they are not identical. They start to differentiate and take on totally different jobs. The outer wall becomes the pigment layer. That's what absorbs stray light to keep our vision sharp. And the inner wall, well, that becomes the neural layer. That's the part that's actually going to do the seeing. And that inner layer becomes this incredibly complex bit of circuitry. You get your light detectors, the famous rods and cones. They pass the signal to connecting cells, and then those pass it to the ganglion cells. And the long tails of all these ganglion cells bundle together to become the optic nerve, the massive cable that sends all that visual info straight to the brain.
So while the retina is busy building all that complex wiring, the lens is doing something that's simpler but just as elegant. It starts as a hollow ball, but then cells from the back wall start to stretch and grow forward, completely filling the space until it becomes one solid, perfectly transparent structure, the eye's powerful focusing element.
All right, we are in the home stretch. We've got the core components. Now we just need to build the rest of the eye's architecture around them. So how does the surrounding tissue know what to do?
Well, this whole developing structure is surrounded by a type of tissue called mesenchyme. The best way to think about it is like biological clay. It's just a bunch of unspecialized cells, a blank slate, just sitting there waiting for instructions on what to become.
And depending on where it is, this clay gets totally different instructions. The mesenchyme at the back of the eye gets told to form the tough protective white layer, the sclera, and the layer with all the blood vessels. But the mesenchyme at the front, it becomes the clear cornea and the tiny muscles that control our focus. Same material, totally different outcomes.
And now, it's just final assembly. The big empty space inside the eye fills up with a gel to give it its shape.
A temporary artery that was supplying blood during development is no longer needed, so it just degenerates and disappears. And finally, folds of skin grow over the front to form our protective eyelids. And just like that, the construction is complete.
So, when you step back and look at the whole process, it's just incredible. To go from a simple, flat layer of cells to this unbelievably complex, light-sensing organ, it all happens through this perfect ballet of folding, signaling, and transforming. It's an amazing story of biological engineering, and it's all designed for one beautifully simple purpose: to let the light in.